Thank you so much, Kofi, for the very kind introduction, and thanks a lot for organizing this very nice event. It's a pleasure to be here. So today I'm going to talk about some of our recent work in the past 3 or 4 years in developing intelligent neural interfaces that could hopefully treat chronic neurological and psychiatric disorders. So I try to specifically highlight how I think the integration of AI and circuits can advance next generation neural interfaces. So I know that we have quite a few students working on this topic in the audience, but just to make sure everyone is on the same page, I'm going to give a brief introduction. So neural interfaces are devices or systems that can record brain activity at various spatiotemporal scales. So we have different types of electrodes for this. So you can, for example, record spiking activity using these microelectrodes, so the activity of individual neurons. Or you can also record lower frequency electrocorticography from the surface of the cortex, or, for example, local field potentials through the DBS leads. So these are lower frequency signals that you can record with these two types of electrodes. Then at the core of this system, usually there is a neural interface chip, an ASIC, which can record and process neural signals, and then it can also deliver targeted electrical stimulation to specific electrodes in the brain in order to modulate the brain or to suppress a symptom or, for example, to provide a sensory feedback. So we call this a bidirectional or a closed-loop system. Essentially, you can read and also write to the brain. So that was an intro, and the challenge that we are trying to address in our Integrated Neurotechnologies lab is how to design more effective and more efficient neural interface systems for a wide range of neurological and psychiatric disorders. And this involves both neuromodulation systems and brain-machine interfaces. So I will talk about both in this talk. So there are different ways to tackle this problem. So many in this field are working on this, but our approach to this problem is interdisciplinary. We essentially combine integrated circuits with algorithms and AI, and we work in close collaboration with clinicians. So our goal is to develop a next generation neural interface circuits that are extremely low power and miniaturized. At the same time, intelligent systems that are safe and effective for chronic use. So in this talk, I will specifically focus on the aspects of designing high density and intelligent systems, but we also work on other aspects of neural interfaces that are shown here. In the past few years, both across academic research institutes and also in neurotech companies and startups, we have seen a trend toward integrating higher number of channels, building higher channel count systems. Here you can see some examples like the NeuroPixels probe, as well as the high-density ECOG by Precision Neuroscience, the polymeric threads by Neuralink, as well as the microwire bundles by Paradromics. So these are all systems with thousands of electrodes, and many of these companies promise scaling up to even tens of thousands of channels. And this means this is a very exciting time for neurotechnology. So this can promise a lot of progress and new opportunities in this field. But this also highlights the need for further miniaturization and designing lower power systems. In fact, much lower power compared to what we have today in order to develop human-grade devices so that we can minimize the heat dissipation near neurons. And the other important aspect is the integration of AI and on-chip intelligence, which could enhance the therapeutic potential of these devices, but this is something that is still missing in many of the current systems. So here you can see a number of neurotech startups that have been developed in the past decade or so, and many of them are actually targeting, different, some therapeutic and diagnostic applications, but the trend that I mentioned toward building higher number of channels is something that we see among these startups that are developing invasive neurotechnologies. There are a number of non-invasive companies as well, but the trend toward building more channels is more among invasive devices because of the higher resolution that you can capture within the brain. And you may ask why we need more channels. Integrating a higher density of electrodes can enable a more accurate detection of disease symptoms, for example, in the case of epilepsy, or more rapid also detection of underlying neurological conditions. In the case of stimulation, when we have these leads with electrodes to stimulate the brain, having a larger number of smaller contacts can reduce the side effects of stimulation, as we can avoid the stimulation of larger irrelevant regions in the brain. In the context of brain-machine interfaces or motor prosthetics, having a higher number of channels can improve the decoding accuracy and enable more natural prosthetic control. So this was the state of technology, but if we look at the devices that are available today as a therapeutic option for patients, there are actually very few examples. So deep brain stimulation, or DBS, for Parkinson is a well-known device. And in recent years, we have seen a trend toward building closed-loop systems, so devices that can record brain activity, for example, like the NeuroPace device for epilepsy, and use that as a feedback mechanism to control stimulation. Another example is the vagus nerve stimulator, which is controlled by the cardiac signal, and the Medtronic Percept device, which is an investigational system for closing the loop in Parkinson's disease to control tremor. More recently, we have seen a couple of other companies trying to build closed-loop stimulation, again, for movement disorders. And in the field of brain-machine interfaces, which we can call this BMI or BCI, despite a lot of progress, we still don't have any clinically viable products. So if we kind of summarize the problems or the limitations of current systems, so all these devices have a very small number of channels, so they have a low resolution, and they also have very limited on-device processing, which limits their therapeutic potential. Many of the current systems are still invasive and bulky, and millions of patients with psychiatric disorders, with Alzheimer's and other conditions cannot benefit from these devices because these are primarily targeting movement disorders and epilepsy, which are affecting a relatively small population. So just to give you an example of what we mean by a closed-loop device, for example, for epilepsy. So here you can see the FDA-approved closed-loop device that we have for controlling seizures in epilepsy. So it's called a responsive neurostimulator, or RNS. So it can essentially record brain signals with two electrodes. So one is a strip and another one is a depth lead. And it can use basically 4 of these contacts to detect the onset of seizure. And the way it can do it is by extracting some biomarkers, some characteristics, temporal or spectral characteristics of the neural signal to detect the transition from a pre-ictal or normal state to the ictal state. And once it detects that transition, it can activate a neurostimulator. It can stimulate on one of these electrodes in order to suppress an upcoming seizure. So this is an example of one channel, but you could have multiple of channels that are capturing a seizure activity. And this is important because for more than 60 million patients with epilepsy, almost 1/3 of them are medication resistant. And one of the very few therapeutic options that some of these patients have is closed-loop neuromodulation. So after this introduction, I'm going to talk about 3 example applications where I think intelligent neural interfaces can have an impact, starting with AI-enabled closed-loop stimulation for real-time symptom tracking. Then I'll talk about our work on developing new methods, new paradigms for closing the loop in psychiatric disorders. And finally, I will present some ideas and some of our recent work toward building intelligent brain-machine interfaces. So this is the work that we presented at ISSCC earlier this year. So I'll start with the first topic, integration of AI on closed-loop devices. So the idea here is that, as opposed to the conventional or the more common approach where we basically can digitize neural signals and transmit them to an external system, an external decoder to detect, for example, symptoms and send commands to activate a stimulator. What we can do is that we can directly integrate the biomarker extraction and machine learning processor on an implantable system in order to detect the onset of disease in real time and activate stimulator. And this has a number of advantages. So it can enable effective intervention with low latency. And this is extremely important in these closed-loop applications. It can reduce the telemetry power and also enable more or better security and privacy for the patients. And thanks to the power of AI in decoding or predicting different neurological patterns and disease symptoms, we can use this in different applications. So you can use this framework to decode or to detect symptoms in disorders like epilepsy, in movement disorders, even change of mood in psychiatric disorders, and so on. And you could also use this to decode movement in a brain-machine interface and use that to control a prosthetic device, activate a spinal cord stimulator, enable a sensory feedback, and so on. So this is the general framework, and in the past few years, we've seen several neural interface ASICs that have integrated machine learning for applications such as seizure detection in epilepsy. So you can see some of the work that are published in the ISSCC conference, but there are many other examples. So if you look at these systems, actually, many of them are focused on the epilepsy application, where you try to detect the onset of seizure and stimulate, and part of it is because of the availability of data in this application. So what I'm going to do in the next few minutes is that I'm going to talk about our neural tree chip, where we wanted to increase the number of channels in a closed-loop system to enable higher density recording, so toward the same goals that I just mentioned, and also extend the application of our closed-loop system to disorders beyond epilepsy. And we wanted to do this while maintaining a very small area and power. So you see that the area per channel for our design is actually quite small. This is an overall block diagram of our neural tree chip. So this system can record high-density ECOG or LFP, so with these electrodes, from up to 256 channel electrodes, and then it can deliver stimulation to selected electrodes depending on the output of the on-chip classifier. So this system is also quite modular and versatile, meaning that we can use this in multiple applications that are shown here. So the idea is that we wanted to overcome the limitations of previous systems. For example, to address the low channel count in current designs, we proposed a highly area-efficient 256-channel analog front end. To extend the application of our SoC to other disorders, we integrated a broad set of neural biomarkers and a multi-class on-chip classifier. And to improve hardware efficiency, we did extensive circuit algorithm co-design and developed an energy-efficient on-chip classifier. So this is the overall block diagram, and I'll talk about some aspects of this in the next few minutes. So one idea that we propose here is what we call channel selective inference, meaning that when we want to train our model, our chip, we want to be able to use high-density data from up to 256 channel electrodes. So for that, we activate 4 modules of 64-channel amplifiers during training. But when we do the inference, we only activate, so we dynamically activate or select 64 channel out of these 256 electrodes, and that means we can basically turn on only one module of the amplifier and save significant power consumption. And the reason we can do this is that usually we observe that in these classification tasks, only a subset of electrodes contribute to the disease-relevant activity, and we can learn that during training. But at the training phase, we need to be able to record high-resolution data. So you can see that in this channel importance analysis that we did on seizure detection from a patient with epilepsy, where we can see that only a number of channels are important in seizure detection. So one challenging aspect of this design, as you can imagine, was to design the analog front end, because we wanted to integrate a large number of channels in a very small area. To do that, we chose to implement a time division multiplexing architecture rather than designing a single amplifier per channel. But the problem is that the electrode DC offsets, so the offsets at the electrode tissue interface, this can actually vary significantly between different electrodes. And this actually means that in our TDM design, we have a very short time in the order of several microseconds for each channel to cancel the DC offsets. And this is even worse in our design because we have this dynamic channel activation, meaning that the offset can actually change between successive feature extraction windows, as you can see here. So for each window of, for example, 1 second of feature extraction, we have a new set of channels and a different pattern of offset that we need to cancel. So to address this problem, we proposed a two-step fast-settling mixed signal front end with a mixed signal DSL, which can enable this fast cancellation. And the idea is that here we have a coarse offset cancellation loop where we basically cancel the offsets in 64 electrodes using binary search. So we store them in a memory. So this is a rapid cancellation that happens first. And once this is done, the neural recording starts with the fine DSL enabled to cancel the residual offsets. So by doing this, and all the modules are here time division multiplex, by doing this we can achieve a reliable recording of neural signal even in the presence of offset change between successive feature extraction windows. But if we only enable the fine DSL as in conventional designs, actually the front end fails to capture part of the neural activity. So our amplifiers are extremely low power. You can see the power per channel and also the area, meaning that we can even further scale this up and integrate a higher number of channels. Another idea that we propose here is the basically integration of a broad set of neural biomarkers that are relevant to common signal classification tasks, such as seizure detection in epilepsy or detection of tremor in Parkinson's disease, as well as detecting finger movement or classifying different finger movements in a brain-machine interface. So we have features in the temporal, spectral, and phase domain, such as, for example, so the spectral energy features, which are kind of common among different disorders. We have features like line length that are relevant to epilepsy, local motor potential for movement decoding, as well as features that are relevant, for example, for Parkinson's disease. And the hardware implementation of these features is also using TDM, and we share major hardware blocks between different features and use some feature approximation techniques to reduce the complexity of implementation. So I talked about the seizure, the application in epilepsy for closed-loop stimulation, but I want to explain also why we need closed-loop deep brain stimulation in Parkinson's disease. So we know that the conventional open-loop deep brain stimulation, or DBS, is quite effective for movement disorders such as Parkinson and essential tremor, but it also has some side effects, such as speech and gait impairment, and changes in mood because of the continuous stimulation, as well as the high energy consumption and the need to replace the battery. So the concept of adaptive DBS tries to overcome these limitations by real-time activation of stimulation based on a feedback from patient's neural activity. So the way that it works is that basically you extract a biomarker, a common biomarker, which is basically the energy in the beta band in 13 to 30 hertz from the local field potentials that you record through a DBS lead. And by thresholding this beta band activity, you can basically trigger your DBS to deliver stimulation in a closed loop way. So a number of studies that are actually pioneered by the group of Peter Brown at Oxford have shown that this method of stimulation can improve motor score and motor symptoms and result in less speech problems compared to continuous DBS, meaning that adaptive DBS is actually a more effective and also more efficient method of stimulation. But the way that it is normally done, as you can see here, is through a single thresholding the beta biomarker, beta band activity, which is actually known to be a very imprecise or not quite an accurate biomarker of tremor. So in a work that we published in 2020, actually we proposed a machine learning framework to more accurately detect the onset of tremor in Parkinson's disease. So this was also in collaboration with Peter Brown, and we showed that by using the data from 12 patients with Parkinson and using a gradient boosting algorithm, we were able to predict the onset of tremor from LFP signals that is recorded through the DBS lead. So you can see the acceleration signal here used for labeling the LFP signal and the output of our decoder. And here we looked into a set of biomarkers of tremor, and interestingly, we found that many simple statistical features, like, for example, George parameter, which kind of measures the variance of the signal, are more correlated compared to beta power, which is more commonly used in these studies. So this kind of informed the choice of biomarkers in the chip that we later designed. So the design of machine learning algorithm for our chip also took several iterations. Earlier in 2018, we published our gradient boosting chip, which is an ensemble of decision trees. And later in this work, we proposed a probabilistic oblique tree, or neural tree, which is essentially a tree-structured neural network. So we have neural networks within the nodes of the decision tree and kind of benefit from both models. And here we also proposed an energy-aware training approach, meaning that we are basically using the power consumption of features in order to regularize our objective function. So what we are doing here is that we are trying to penalize those features that consume more power. For example, phase-based features or spectral features compared to temporal features are more complex. So we are essentially encouraging our model to use a higher number of low complexity features to reduce power consumption, and this is with a very minimal or marginal reduction in performance. Our probabilistic model is also compatible with gradient-based optimizations, meaning that we can use aggressive pruning and quantization to reduce the size of on-chip memory. So here you can see this dynamic channel selection that I mentioned earlier through the decision path of the neural tree until we reach a dominant class. So the hardware implementation also is quite simple. So we have a single MAC and comparator, which is doing all the feature weight multiplications in a TDM fashion. Finally, we also integrated a 16-channel high-voltage compliant neurostimulator on the same chip. And here we use the 4-phase charge pump with high-frequency clocking in order to reduce the area per channel. So we achieved almost 3 times the smaller area in a standard CMOS process. So we verified our chip with multiple publicly available data sets. As you can see here, for example, we use the CHP-MIT EEG data set for epilepsy, as well as the intracranial EEG data for verifying the seizure detection performance. And we use the data from the Parkinson patients that is recorded through DBS lead to verify the tremor detection. So you can see examples of how the chip can actually classify the input neural signals and the overall performance. We also tested our system in vivo in collaboration with the group of Stefani Lacour at EPFL, who developed these soft micro-ECOG arrays. So we implanted two arrays of 16-channel electrodes in the cortex of an epileptic rat, and we were able to validate different functionalities in vivo. So for example, you can see this changes in temporal and spectral features at the onset of seizure. So if I compare this with previous systems, we were able to significantly increase the number of channels. So this is the highest density closed-loop system that we have today. We were also able to reduce the area per channel, as well as improving the energy efficiency of the system. And this is despite the fact that we integrate a broader set of neural biomarkers that actually improve or increase the flexibility of system to be used in different applications. So we are also working on the machine learning algorithm design aspects of closed-loop systems. So for example, we are very interested in models that can dynamically exploit both the temporal and the spatial characteristics of EEG signal in order to improve the accuracy. And we've had very interesting results on detecting the focal as well as generalized seizures. So we have a paper in ICML in review only on the algorithmic aspects of it. So this concludes the first part. And in the second part of my talk, I'm going to talk about neuromodulation devices, so developing neuromodulation systems for psychiatric disorders, which, as we all know, are affecting millions of patients around the world. So you can just see some statistics here. So depression is one of the leading causes of disability, and the prevalence of disorders continues to increase. It is a major economic burden as well. And really the sad part is that many of these patients are out of options. So they try all kinds of medications, different therapies, and nothing works. So we urgently need new therapies, such as device-based therapies for these disorders, but surprisingly very little has been done for this application. So the conventional open-loop DBS that I mentioned, and it is effective for Parkinson, it has shown some promising results in patients with mental disorders. So different brain regions have been studied. But it also has shown some mixed results, and there has been variability in terms of efficacy across different trials. So this means that it is not sufficient to become a clinical device for depression. So the reason that this open-loop DBS actually cannot be very effective for mental disorders could be because of the fact that these psychiatric disorders actually involve multiple distributed networks in the brain. So they are quite complex to tackle. So if we think about current therapies, so the basic problem is that these treatments actually are not targeting the mechanism of disease, and they do not show any proof of target engagement. And this is because these psychiatric disorders actually arise from communication failure between multiple distributed brain regions. So a solution based on a focal single region stimulation like we do in Parkinson may not be an effective therapy for mental disorders. So we have been working on a project that is funded by the US National Institute of Mental Health, and this is in collaboration with Dr. Alec Veach from University of Minnesota, who is a psychiatrist. And our goal here is to develop a new neuromodulation system that can deliver a new mechanism to control and potentially even correct the disrupted communication to treat mood and anxiety disorders. So one idea here is what we call phase-locked stimulation, meaning that we can stimulate one region in the brain at target phases of neuronal oscillation in another region. And there's increasing evidence showing that this method of stimulation can result in more physiologically meaningful effects in a long-lasting way because it can actually improve the plasticity. So there are many groups that are actually trying this method with non-invasive techniques such as transcranial magnetic stimulation. Another idea here is measuring the cross-regional neural synchrony. So the different brain regions actually communicate through these neuronal oscillations. And in disorders like depression, and even in Parkinson's disease, the degree of cross-regional communication is a key indicator of pathological brain state, as you can see here. So if we could have biomarkers or metrics that can measure this cross-regional synchrony, so 2 examples are shown here, such as phase locking value and phase amplitude coupling with the definitions that are shown here. So if we could measure this connectivity or the level of synchrony between different brain regions, then we can use this to control or to use our phase lock DBS to regulate that synchrony and improve symptoms in psychiatric disorders. So this is another feature of our device that is able to extract these network-based biomarkers in order to regulate the symptoms in mood disorders. So based on these ideas, this is the block diagram of the first prototype device that we have developed for this. So here we have electrodes that are implanted in 2 regions of the brain. So it can be either deep brain electrodes or other types of electrodes. And then we have 16-channel low-noise amplifiers. And the output of ADC is then sent to bandpass, an FIR filter for bandpass filtering and Hilbert transform to extract the phase and amplitude. And basically, this is to extract the real and imaginary parts to extract phase, which I'll talk about it later. And then we use that phase and amplitude envelope to calculate these synchrony biomarkers, such as phase locking value and phase amplitude coupling. And we have a flexibility to use one or multiple biomarkers and threshold them in order to activate a 4-channel on-chip neurostimulator. So the key idea that we developed here was reducing basically the complexity of phase extraction, because this is the key function that we need in our system in order to enable phase lock DBS as well as extracting the phase-based biomarkers. So to do that, we proposed a lightweight phase extractor, which is based on a first-order arctangent approximation. So essentially, we represent the phase in each quadrant by the fraction of real and imaginary parts with an offset value. And in order to cancel the error that is caused by this approximation, we use a small lookup table to reduce that error. So with this method, we are able to extract the phase with sub-LSB error for different combinations of real and imaginary part. And here we also verified our system in vivo, so you can see that the phase extracted by our LPE can correctly track the phase that is actually the ideal phase extracted in MATLAB using the arctangent function. And our proposed methods can significantly reduce the area and power consumption compared to conventional CORDIC implementation. And once we extract the phase of neural signal, we can use that to extract these biomarkers of phase like PLV and PAC. And here, again, we have some shared hardware modules that we can share between these 2 features, and we use some approximation techniques to reduce the complexity. So here you can see the output of our SOC. So this is, for example, PLV and PAC that is measured by the SOC. So this includes the whole front end and the biomarker extraction, which is closely, again, tracking the ideal features that are extracted in MATLAB. So I'll explain in the next slide that how these are actually, how we did the in vivo testing. And this, again, results in significant reduction of power consumption compared to the previous CORDIC implementation. So we extensively validated our system in vivo, and for this we implanted 2 arrays of microwires in the infralimbic cortex, or IL, and the basolateral amygdala, or BLA, which are involved in fear expression in the brain. So these are important, for example, in anxiety disorders. So in addition to the biomarkers, we verified the closed-loop functionality of our system in 2 different stimulation modes. So in the first mode, we are stimulating at certain phases of theta band activity in the IL region. So you see that the stimulation is locked to 180 degrees phase of theta oscillations in the IL region. And in the second example, we are locking the stimulation to both the phase of, for example, IL theta, as well as the PLV. So we are only stimulating if this PLV, which is a measure of, again, connectivity in the brain, is within a certain therapeutic window, within a threshold range that could be programmed by a physician. And once this is within that range, we deliver these stimulation pulses at those points that our phase is at the target value. We also have an option to deliver or to generate a pseudo-random phase threshold to enable a basically random disruption or intentional disruption of synchrony to treat certain psychiatric conditions. So sometimes we don't want to increase the synchrony. We may want to actually reduce the level of synchronization in the brain. So here you can see the dye photo. This is a 65 nanometer process, and the active area is 2.24 square millimeter, and the total power consumption is 60 microwatt. So the contribution of this design is that it significantly reduces the power consumption for extracting these important biomarkers. And this can enable us to integrate a larger number of these biomarkers when we want to look into multiple, distributed regions with higher number of channels. And this is the first chip that enables phase lock deep brain stimulation with flexible stimulation modes to treat psychiatric disorders potentially that disrupt communication between brain regions. So we have been doing extensive in vivo validation of the system and basically planning for first-in-human testing in patients with treatment-resistant depression once we integrate our chip with an unimplantable stimulator. And in the future, one idea that we can add to the system is actually we can use a machine learning-based decoder to guide the output of this or to guide the phase locking DBS. So we can use this to, for example, detect the state of mood and control phase locking DBS. So this is an idea that we presented or discussed in our recent CICC paper this week. Okay, so that was the second topic. And finally, I'm going to talk about developing intelligent brain-machine interfaces. So these are devices or systems that can record brain signals. So usually we have an array of electrodes. Very common is the Utah type, or you could also have an ECOG array. Then following that, we do some processing on the signal. So you would extract some task-related activities and then decode movement intention. So like many other fields, in recent years, AI has made an enormous impact in the field of BCI. So you can see some breakthrough examples here that enable, for example, control of robotic arms. Here you can see that you can even restore walking ability in patients with a spinal cord injury through decoding of their movement intention using a brain implant and sending that to a spinal cord stimulator to restore their walking ability. We have even seen systems or efforts to decode the handwriting, so the attempted handwritten characters from brain activity to convert that to the text. So this is called the brain-to-text BMI. And finally, there are even systems that can decode the speech ability and restore the communication for patients who have lost their ability to speak. So these few examples, especially these three ones, were, I think, a turning point in the development of BCIs in recent years, which actually show the great potential of these technologies. And these two examples enable much faster communication speed compared to any BMI that existed before. And this is really a groundbreaking work that shows you can even, build a bridge between your brain and the spinal cord and use that to basically help patients regain their walking ability. So these are at the level of software, or as you can see with bulky wired commercial systems, which as we all know, this can result in limited mobility for the patient. This is not ideal. This requires basically power-demanding computers. And the goal is that hopefully we want to see that in the future, these BMIs would be fully implantable and miniaturized that could enable more convenience and safety for the patients. And we will also want to enable intelligence on these systems so that we can ensure high decoding accuracy over long periods of time. So in terms of circuit design for BMI, in recent years, we have seen a number of systems that have been mostly focused on spike sorting algorithm. So this is the process of isolating different spike patterns. But as we know, spike sorting is not sufficient for decoding movement intentions. There are a handful of systems that integrated on-chip movement decoding, as you can see here, using different algorithms. But all of these systems actually focused on very simple BMI tasks, such as decoding of finger movements. This is the case also for our neural tree chip. And similarly, the task of spike sorting is actually a low complexity task. It involves a small number of classes. But on the other hand, movement decoding deals with much higher dimensional data with higher number of classes and more degrees of freedom. So this means we need more complex models compared to, for example, clustering techniques that are used in spike sorting. So in addition to the performance, we also need to ensure efficiency of hardware and also scalability so that we can even integrate higher number of channels in the future. So toward building a BMI chip, we were interested in these two types of systems that actually attempt to decode handwritten characters or speech in patients with paralysis. So basically the idea here is that, so the reason these systems outperform previous BMIs is that they attempt to decode the rapid sequences of dexterous movement behavior, such as handwriting and speech, and that results in much faster communication recovery, communication, and faster speed compared to previous BMIs, because these behaviors are fundamentally easier to decode compared to point-by-point movements that was done in previous BMIs. So we focus on the first system, the brain-to-text BMI. So in the original work from Shenoy team at Stanford, they actually used the neural recordings from 2 Utah array implanted in the premotor cortex. And they used the recurrent neural network to achieve a 94% accuracy for decoding 31 characters. And they showed the highest speed BMI that was reported till that date. But in their approach, they actually used a complex RNN model, which has a very large number of parameters. So obviously, this wouldn't be very, a good idea to implement at the chip level. So motivated by this design, we wanted to propose a more hardware-efficient solution. And the idea that we proposed is, this is actually inspired by the visual processing mechanism in the brain, by the way that our brain selects visually salient objects. So when we look at an object, our brain actually generates these saliency maps, which highlight the most visually salient objects, like this one, and basically draws our attention toward that. So in a similar way, we extract neural code. So this is a signature of brain activity that highlights the most distinctive features for each class, and we can use that in the decoding process. And the advantage is that this enables effective extraction of information. It's highly interpretable and results in significant reduction of dimensionality. And it also allows us to use a low complexity decoder with much smaller number of parameters. So here I'm also showing a visualization of neural activity for different characters. So this is for the characters of H, E, L, and O. And the process of extracting this distinctive neural code. So we call them DNC, or distinctive neural code. So our model here actually extracts these class saliency maps for each character, which basically represent the discrimination of a specific class from others. In other words, this shows the importance of different features for decoding, and then our model selects those features with the highest class saliency value for the decoding. So this is based on a class saliency metric that I'll explain later. So based on this here, we can see this clear separation of neural activity for each character in a three-dimensional DNC subspace. So this is only using 3 DNC dimensions. And we have that for each character. So we built a complete system using this DNC-based decoding approach. So in our system, we also use the same data from Shenoy Lab. So this is pre-recorded data from 2 Utah arrays, 192 channels. And following some pre-processing and smoothing of the spike rates, we actually send that to our DNC-based decoder, which is compressing the data into a low-dimensional DNC space, which is now basically can be decoded with a low-complexity decoder. So in this case, we use a linear discriminant analysis or LDA classifier. And we have significant reduction of dimensionality in each processing step, and over 150 times more dimensionality reduction compared to the previous BMI, which used spiking band power features. And thanks to this enhancement of information, we achieve a performance of 91.3% from our chip, which is comparable to the 94% by the software-based RNN model. And this is despite the significant reduction in dimensionality and the simplicity of our approach. So here you can see the block diagram of the entire system. We also implemented 192-channel neural recording chip just as a proof of concept to show that we can actually integrate the recording and decoder chip. So with the previous systems, the majority of them only had the decoder chip. And then we have in the decoding chip, basically so it can receive up to 512 spike rates as input, which are then smoothed by basically, they are convolved by a Gaussian kernel to reduce the sensitivity to temporal misalignment. So this is imported in the decoding process. And then the onset of task-related neural activities are detected, and the spiking rate is basically aligned with that onset. And in the next step, we extract the DNCs based on the timing information, as well as the index that we train and send it to the chip. And finally, those DNCs are are sent to the LDA classifier to calculate the distance between the DNCs and the hyperplane in order to generate the output class. So here, for in every second, we have we basically repeat this decoding process every second, meaning that we have a decoding speed of 60 characters per minute. So here you can see a bit more details about the training algorithm, but the key idea here is that we use this class saliency metric, which kind of measures the basically magnitude and also the homogeneity of the between class distances. So this kind of shows how uniform the distance between multiple classes are in order to extract the importance of features and basically extract these DNCs with highest saliency value, which are then used in the classification process to output one of the 31 characters. So basically, 1 second after detection of the activity onset, we have one character as predicted output. So our DNC-based hardware results in significant reduction in hardware complexity in terms of number of MAC operations and memory reduction compared to an LDA that we would use without DNC. So if you would use LDA without applying the DNC extraction. And thanks to the sparsity of these DNCs, we can also share the memory across different channels and further reduce the memory. You can see our chipset here. Again, this is in 65 nanometer process. This is the neural recording chip, and this is the decoding chip. The active area of the entire design is 2.46 square millimeter, and the total power consumption is 883 microwatt. The decoder chip only consumes 223 microwatt, and one reason for this low power consumption is that this is an activity-driven decoder. We only activate our decoder upon detection of those activity onsets, and otherwise this is disabled, which results in power reduction. So here you can see the measured results from our chip. So here you can see the basically the original or the labeled characters. So these are attempted characters by the patient, and then you can see the output of the decoder chip. We also have the confusion metrics here for the decoding probabilities, and we tested this across 10 sessions, same data as the original paper, and achieved a decoding performance of 91.3% from the chip. So this is for the software model of the chip, which is comparable again to the original work. And this is much higher than using a correlation-based method. So using only 100 DNCs, we achieve a much better performance compared to using more conventional methods. We also tested our system in vivo. So for this, we used the NeuroPixels probe, which was implanted in the auditory cortex in a rat. So this is an auditory stimulation task, and we wanted to classify 6 different frequency bands using our decoder chip, and we achieved an accuracy of 87% for this classification task. We also verified our recording chip, so we were able to record, for example, spiking activity on different channels of our amplifier. So to conclude, I think the contributions of this system is that we were able to significantly increase the task complexity. So this is the first on-chip brain to test BMI, and we were also able to increase the number of channels while reducing the area and power by at least 1 order of magnitude. I think this is just one example showing that by integrating algorithms and machine learning and signal processing techniques, we can really go beyond simple BMIs and enable much more opportunities in the future. So to summarize my talk, I covered 3 different types of neural interfaces, and I hope I was able to convince you that the future of this field is moving toward higher intelligence, better efficiency, miniaturization, and higher resolution. We are going to see more and more channels in these implantable systems. And finally, I'd like to thank my students, our collaborators, and funding sources. And I want to mention that I have several postdoc openings in my lab on really interesting projects related to neural interfaces. So if you're interested, please contact me.